Okay, thanks, Diego. Hopefully you've all had a drink and a pastry, and the cool temperature in here will keep you awake during the next session of talks. So I'm going to talk to you today about why do dinosaurs have different shaped skulls. And the obvious answer to that, of course, is to do different things. Dinosaurs were a very successful group, of course. They were around for over 150 million years, and this variety of different skull forms that they have were involved in things like we've just seen in Victoria's talk in terms of signaling and fighting, but also in terms of allowing these animals to exploit multiple different dietary niches, and also to be able to exploit different types of habitats, things like tooth batteries, development of unique types of tooth enamel, and potentially also very unique kinetic mechanisms in their skull. But what I'm hopefully going to talk to you about over the next 20 minutes or so is that although this kind of diversity in skull form is iconic really in how we understand dinosaurs, we actually don't understand very much in terms of the macroevolutionary sense about how this diversity was achieved, to a certain extent when it was achieved, but also as well how really this variety in form that we see really links to the different types of functions that these skulls were undertaking. We may expect there to be kind of a one-to-one relationship between the shape of the skull and the function that it undertakes, but as you saw from Carl's talk earlier, there's all these added complications about how we interpret soft tissues. And on top of that as well too, there's the potential constraints that we find with phylogeny. Do skulls simply look the same because animals are related to each other? And other types of constraints as well. So with allometry, these animals are achieving massive differences in size. What role does allometry play in controlling form? And other types of constraints to do with how the skulls are constructed during development. So what I'm going to present to you is to kind of the current state of play where we are in understanding the quantification of skull form and the quantification of function, and our understanding of the relationship between those two entities, and really therefore where can we go next from this standpoint. So if we start with characterizing variation in form, this has been achieved to some extent through the use of two-dimensional geometric morphometrics. So the method in which you take a shape and you characterize it by a series of landmark data points and then look at the variation in that shape. And so one of the first studies to do this was by Steve Brusaci. Steve is talking to us all later on this afternoon. And Steve and his colleagues looked at theropod cranial variation, and they did this looking at a lateral view of the skull of a group of theropod dinosaurs. And they found that nearly 65% of the variation in form can be characterized by certain trends in changes in shape. So for example, an anterior-posterior lengthening and shortening of the skull was very pervasive in terms of directing the changes in shape, and also a kind of taller and shorter orbit and a deeper cheek and changes in the depth of the skull as well. And they found as well with this study that there was a strong phylogenetic signal in this data set. So basically skulls that are animals that were quite closely related to each other tended to have quite similar shaped skulls. But there was some convergence towards similar skull shape in different groups of large-bodied theropod dinosaurs. Around the same time, a similar study came out by Foth and Rauhut, and they also did a two-dimensional geometric morphometric study on the lateral view of the side of the skull. And although they used similar taxa, their landmark data set was slightly different. But as you can see perhaps from this plot here, they also found kind of similar trends in that they also got a strong phylogenetic signal in their data set. They did find, though, that carnivores and herbivores could be significantly distinguished from omnivores and herbivores in their skull shapes. But you couldn't really tease apart omnivores and herbivores in terms of the difference in their skull shape. Now, what was interesting about these two studies is if you consider them side by side, although they use different landmarks, they're broadly giving the same shape signal, in that we're getting the clustering of different groups in different regions of this shape space variation. And both of these plots are also showing this convergence towards similar skull shapes in different groups of large-bodied theropod dinosaurs. Some other studies have also looked at other groups of dinosaurs. So this was a study by Young and Larven in 2010, and they looked at the variation in skull shape in sauropodomorph dinosaurs, again in 2D lateral view, and then also in a two-dimensional dorsal view as well too. And I've just color-coded these plots here so you can see a kind of paraphyletic assemblage of prosauropods in the blue, and then the sauropods in the orange color. And this study also found that there was quite a phylogenetic partitioning in the data. So sauropods tended to have similar shaped skulls to each other. Prosauropods, albeit a paraphyletic assemblage, also had similar shaped skulls. And this taxon that tends to sit in isolated space here is one of the most earliest sauropod dinosaurs, Shunosaurus. Now, a study that we did in my lab here looked at, instead of skull shape variation, we looked at jaw shape variation. And this was done for 167 ornithischian and sauropodomorph taxa. So this was the shape of the mandible. And what we found from this is that we did find that there was a significant difference in the shape of the jaws between sauropodomorphs and ornithischia. And what we also did was tracked this variation through time as well. So if we start off here in the late Triassic, we can see that we have a few sauropodomorph taxa, and then the very earliest basal ornithischia. And as we move through to the early Jurassic, we can see how this variation in jaw shape is being exploited. And where these points start to overlap, that's where we're seeing similar jaw shapes being possessed by different groups of herbivorous dinosaurs. So there's some overlap here in the early to mid-Jurassic between the jaw shapes in the prosauropods, the basal ornithischia, and in the earliest thyreophorans as well too. And likewise for the late Jurassic as well too. These animals seem to be converging in similar shapes. But as we move through into the Cretaceous, then we see a real divergence of shape space here. And these different groups of herbivorous dinosaurs are occupying different regions of this shape space, meaning that their jaws are basically diverging in shape. And here we move through to the later part of the early Cretaceous, then into the late Cretaceous, and then finally 85 to 65 million years ago, where these different groups are occupying these different types of jaw shapes. So again, what we're seeing here is a fairly strong phylogenetic signal. So that seems to occur across all of the clades that we've looked at: theropods, ornithischia, sauropodomorpha. Animals tend to have similar shaped skulls or jaws because they're closely related to each other. But perhaps within that, there is an allometric signal as well too. Particularly in these large-bodied theropods, they seem to converge towards a similar type of skull shape. And this may be linked to dietary ecology because again in theropods we see some teasing apart of different skull shapes for herbivores and different skull shapes for carnivores. So, where does this leave us then in terms of characterizing function? So there are multiple ways in which we can do this. We've seen great examples of this in some of the previous talks before the break. Some of the things that I've highlighted here are ways in which we can actually start to quantify and characterize function in extinct animals. One of these is looking at the muscles themselves, as we saw from Carl's talk, looking at things like muscle leverage, calculating something called the mechanical advantage, which is basically the efficiency of the leverage of the jaws in this context. We can also look at the output that muscles can generate in terms of bite forces within the skull. We can do quantitative trait analysis as well too, taking a series of traits which we deem to be functionally important and seeing how they plot out in multivariate space. So similar to the shape analysis, but where taxa plot closely together, they share similar functions. And then also mechanical analysis as well too, looking at structures as beams and seeing how they perform, or doing stress and strain analysis to see how structures deal with mechanical loads generated during function. So I'm not going to talk anymore really about quantitative trait analysis or beam analysis, and instead I'm going to focus on what we can get from the fossils in terms of muscles and mechanical analysis. So muscle reconstruction, again in Carl's talk we had an introduction to the principles behind this, the fact that we can use living animals which bracket phylogenetically our extinct taxa of interest, where we can gauge what the real muscles look like, and particularly when those muscles attach into bits of the skeleton, what signatures or scars do they leave? These so-called osteological correlates then we can look for in the fossils of extinct and the preservation of extinct animals. So where we see a ridge or a fleshy, smooth surface that in a fossil that's in the same place in a close bracketed phylogenetic relative, we infer that that same muscle or that same tendon is present in our fossil. And this has been done with great detail in many different groups, or a number of different groups of extinct animals, using this osteological correlate and extinct phylogenetic bracketing approach. And ultimately, what all of these studies have shown, if you consider them together, is that most dinosaurs, even the most derived ones, possess essentially a diapsid skull form. So they retain a fairly small brain, 2 temporal fenestra at the back of the skull. And what that means is that therefore they tend to retain a more plesiomorphic, general archosaurian adductor muscle condition. And that's the same kind of muscle condition that we see in extant crocodilians. And that pretty much is the case in most of our groups of dinosaurs. Once we have a handle, though, on actual muscle reconstruction, then we can look at quantifying things like the leverage potential of those muscles and then the output forces that generate. So muscle leverage we can measure by looking at either the amount of muscle force that goes in compared to the amount of bite force that goes out, or if we don't have those measurements, we can look at the length of the levers that are generating these forces in and out of the skull. So in this case here, we're measuring something called the mechanical advantage, which is the length of the input lever, which is basically this perpendicular distance here from the line of action of our input muscles, over the output lever, which is the distance from the jaw joint fulcrum here to where the animal's biting. And a higher mechanical advantage signifies a more forceful jaw closure. A lower mechanical advantage signifies a less forceful but a faster jaw closure. In terms of muscle volumes as well too, once we have a handle on where those muscles can attach, we can digitally then reconstruct the size of those muscles themselves. And once we have an estimate of muscle volume, we can then calculate how much force those muscles can generate. We do this by measuring something called the physiological cross-sectional area, which is the volume of that muscle multiplied by the cosine of the angle which that muscle deviates from the horizontal, or the fibers deviate from the horizontal, divided by the length of those fibers themselves. And we take this value and multiply it by a value of muscle stress, which gives us a value of muscle force. And those of you amongst you are probably picking out that there are a number of different assumptions that we need to make when we use this equation here about things like fiber length and muscle stress as well too. But taking these measurements then and calculating them for groups of different dinosaurs, how does this then impact our understanding of skull function? So this was a study that came out a couple of years ago, which calculated the leverage of the jaw and essentially the efficiency of converting muscle force to bite force for a group of different ornithischian dinosaurs. And it calculated something called the relative bite force, which in this context is essentially the mechanical advantage, so the efficiency of the leverage of the jaws. And it's color-coded here so that where you have warmer colors, you've got more efficient jaw leverage. And the study basically showed that there are 2 main groups of ornithischia, the hadrosaurs and the ceratopsians, which are evolving towards having these more efficient jaw levers. And it's different for when the animal's biting at the front of the jaws, and other groups are perhaps optimizing more for biting at the back of the jaws. And this tallies with other studies as well too that were done that looked at these jaw leverages in these particular clades. So Mallon and Anderson, for example, also found that ankylosaurs down here within the thyreophorans have a relatively low leverage in their jaws, whereas the ceratopsids and the hadrosaurs had a fairly high leverage, the same as this study actually found here. So despite reconstructing muscles with this kind of general plesiomorphic muscle condition, muscle moment arms and therefore the kind of relative bite forces and leverage are varying between clades, indicating that there's perhaps some kind of functional driver there that's causing this variation. And this was a study by Sakamoto in 2010, which looked at the leverage of the jaws in theropod dinosaurs, calculating a mechanical advantage value across the length of the jaws. And this study also found that there was a strong phylogenetic signal in the data itself. And actually the evolution of mechanical advantage across these different theropod dinosaurs was no different from a Brownian motion effect. So this is a study done by a former master's student of mine, James Ormiston, and in this study we looked at the whole of mechanical advantage across all of dinosaurs. And you can see here that you can basically draw a line almost right across here, which is where you've got theropods to the left and everything else to the right. And so within ornithischia, we get these kind of clades, like in the previous studies, which show increased leverage in jaw muscle advantage. And then in the ceratopsians and in the hadrosaurs as well. But pretty much the whole of theropods relative to the rest of the dinosaurs have got fairly weaker leverage in their jaws. And this is interesting because it gives us a perspective of the whole of Dinosauria, but it also sits at odds with the fact that all of the estimated bite forces that we see for dinosaurs are much, much higher in theropods than they are in any of the other clades. Now notice this is a log scale here, so these values are much, much higher than what we see in other taxa, omnivores, herbivores, and herbivores. So although the theropods have got particularly poor leverage in their jaws, which is perhaps not evolving in any kind of maybe a functionally advantageous way, they are actually showing that they're able to generate extremely high bite forces. So there's a kind of counter effect that's going on there. So we have this plesiomorphic adductor arrangement. Hadrosaurs, ceratopsians have got relatively more efficient jaw leverage. The theropods have got less efficient leverage than the sauropodomorphs and the ornithischia. But those theropod animals are generating the highest bite forces by some margin. So then how does this link into skull mechanics and our understanding of what those forces are actually doing to the stress and strain within the skull? So this was a study that I did a few years ago, which looked at stresses within the skulls of a variety of different theropod dinosaurs. Now the limitations of this study was it was two-dimensional, so it was basically taking the shape of these skulls as a kind of two-dimensional planar shape and saying if they had loads applied to them, how would they stress and strain? And these finite element models you interpret by basically the areas where the blue colors are low stresses and the warmer colors are higher stresses. And for these variety of different taxa here, if we plot them against jaw, if we plot the amount of stress in the skull against skull length here, we find that skull stress increases, but actually it increases with negative allometry, so that these larger taxa have got proportionally lower stresses within their skulls. So perhaps this may be a reason why we're seeing this convergence towards similar shapes in our large body taxa, and there may be some kind of advantages of large size in terms of accommodating stresses within the skull. However, if we look at other studies which have looked at stresses within the skull in 3 dimensions, we see a kind of mixed picture here. So this study compared 3 taxa, Plateosaurus, Stegosaurus, Silicosaurus, of similar-ish skull lengths. And you can see, if you interpret these warm and cool colors, that Plateosaurus here at the top, although not being the largest skull, has the lowest amount of stress within the skull. And Stegosaurus has got high stress when it's biting at the front, low stress when it's biting at the back, and then the opposite in Erlikosaurus here. But then if we just look at the jaws themselves, the jaws are telling us again a slightly different picture, because there's fairly high stresses throughout all the jaws within the different taxa, and there's very little distinction between those different taxa. So we're getting a slightly different signal between the skull and the lower jaws. In this study here, we can compare Plateosaurus again to a larger taxon. We scale these to the same relative size so we can look at the effects of shape. We see lower stresses within the skull here, which is actually in life a much bigger example. And again, this study here as well on the right, we take a large-bodied animal like Allosaurus and we compare the stresses within the skulls of smaller taxa here. If all of these skulls were approximated to the same size, the stresses within them are quite different. Allosaurus Dinosaurs is capable of withstanding much higher stresses than these other taxa, all being considered and size being equal. But also we do see the effects of other things like the expansion of the brain and endocranial development, which is also controlling how the stresses are withstood within the skull. And for taxa that are of similar size, we do see differences due to feeding ecology as well too. So Camarasaurus here and Diplodocus here, although similar sized skulls, when they scale to the same size, they do perform in slightly different ways. And this tallies in with our quantitative analyses, which show that these taxa may actually be functioning in different ways to help partition niches within the same kind, well, it's living within the same environments. So we've quantified form, and there are ways in which we can quantify function, although there's still a long way to go in both of those cases. What about the link between those two circumstances? So these two studies here, which looked at variation in shape, actually the Brusati et al. Study found that there was actually a weak correlation between shape variation and a functional proxy mechanical advantage. Whereas the Foth and Rauhut study found that there was actually quite a strong correlation between skull shape and skull function, when the proxy of function was a measure of skull strength, or my measures of skull stress that I showed you for those 2D shapes earlier on. So again, depending on which functional proxy you look at, there's either a good link between form and function or a not so good link between form and function. In our study of all herbivorous dinosaurs in sauropodomorph and ornithischian taxa, we also looked at functional variation as well too. And we found that the two clades were statistically distinct in terms of their function. But if we looked at the variation or the disparity in morphology through time and disparity in functional traits through time, these two things were decoupled. So when we see a change in the variation in shape through time, we don't see a change in the amount of variation in function through time. So to wrap up then, why do dinosaurs have different shaped skulls? Clearly it's because they're allowing these animals to do different things. But what seems to be coming through is that phylogeny does seem to be having quite a strong effect on form, and that many animals seem to have similar shaped skulls because they're quite closely related to each other. But there is some distinction in that large-bodied theropods seem to converge towards similar shapes, and there seems to be some distinction due to dietary ecology between carnivores and herbivores. We do see differences due to function. We have this retention of this basic kind of simple jaw muscle adductor anatomy, but within clades, the leverage of the jaws varies, but this doesn't seem to directly map onto the ability of these animals to generate large or not so large bite forces. So there's quite a bit of variation there. And also, perhaps large size in some of these theropods is conferring some kind of advantage in terms of accommodating stresses. But the taxa are not only just getting big, they're also partitioning how they use their skulls in different ways. And if we try and look at this relationship between form and function, it depends on what trait you actually use to quantify function. Sometimes there's a tight link between that trait and form, and sometimes there's not that much of a tight link there. And we still broadly lack a real understanding on a macroevolutionary context about how any of this links to diversity as well too. And I think partly that's because the similar themes to as Carl was saying earlier on, it's really difficult to actually quantify and bracket within sensitivity how some of these animals are performing. And we need to get that right first in individual taxa before we then extrapolate it up into looking at macroevolutionary trends. But we're at the beginnings, I think, of trying to be able to do that now. So I'd just like to wrap up and say thank you to all these funding agencies, and a huge thanks to all these people who've passed through my lab as PhD students and master's students, whose work I've talked about in this presentation, and to Paul and Phil, who were collaborators as well on this project. And thank you very much.